It is now my pleasure to turn today's program over to Belinda Lee.
It is my pleasure to introduce Aaron Tyznik. Aaron is a cellular immunologist by training. He has a strong interest in engineering and enabling easy adoption and application of flow cytometry. Aaron is interested in advancing flow cytometry through education to minimize the challenges faced by researchers and also driving advances in the technology to reduce the need for this education. These advances include instrument setup, standardization, design, and addressing the challenges of utilizing fluorescent dyes in panel design. Aaron is a recipient of the prestigious ISAC Mary Lou Ingram Scholars Program Award and ISAC Workshop Presenter. Aaron has extensive experience as a cytometer application and design advisor and is also a high-parameter flow cytometry specialist and scientific liaison. Aaron was a graduate and postdoc NIH fellowship recipient, former lecturer, and graduate-level immunology course class facilitator. Thank you and welcome, Aaron.
Thank you very much, Belinda. So as Belinda mentioned, I'm a manager in the applications element at BD, and I'll be talking about today the utility of a modular regulatory T-cell panel and the application of it in a high-dimensional analysis. And we're going to basically go through the process of designing panels to accommodate all the needs for your particular requirements for your specific assay. And for us, we're actually interested in looking at regulatory T-cell biology and the activation and homing specifically. So as many of you are familiar, we have the usual lymphocytes. Most of us think about typical T cells and B cells, along with the antigen-presenting cells. And within the T cell compartment, we have what we know of as the killer cells, our CD8 T cells. And we also have our CD4 T cells, which we often think of as our helper T cells. Now, the helper T cells are often involved in helping immune responses. They may aid in B cell response. They may help with the CD8 T cell response. And there's multiple functions of all of these different cells. And one that particular cell population that we got very interested in and that I'll be talking about today is the regulatory T cells. The regulatory T cells are often thought as possibly as a peace officer, if you will. They often are involved in regulating the immune response, and they're indispensable in many different situations, whether it's during an active immune response to an infection or if it's controlling an environment such as a mucosal environment to keep everyone happy and make sure that the cells are able to respond appropriately. So when we want to study an area of biology, whether it's the CD4 helper T cells or our NK cells, one of the most useful tools that we have in the laboratory is a flow cytometer. And today we'll be talking about the BD Fax Lyric flow cytometer. And with any instrument, whenever you want to start building a flow cytometry panel, you have to actually understand what your instrument configuration is. This is independent of whatever cell populations I'm looking at. I need to know what channels I can look at so I can pair my antibodies or my dyes and viability stains accordingly. So with a BD Fax Lyric, where we're trying to build our T regulatory panel, I'm going to be working with an instrument that has 3 lasers, a violet 405 nanometer laser, a blue 488 nanometer laser, and a red 640 nanometer laser. And you can see the corresponding fluorochromes that can be detected on this particular instrument. So when I want to think about building my panel, I have to basically make sure that my instrument can basically support all the markers that I want to use in my area of biological research. Well, in this situation, I'm trying to build a T regulatory panel. And when I build a T regulatory panel, I almost always have a set of common core antigens that I have to use to define my cell populations of interest. Again, this is true if you're studying natural killer cells, dendritic cells, and it's often very common that a laboratory has a common set of markers that everyone is looking at, and each individual researcher will add their own markers as it fits for their specific biology. Well, for myself, when I look at my common core antigens for my Treg panel, I almost always utilize CD3 to define my T cells, as well as CD4, because as we know, T regulatory cells are CD4 positive. Additionally, there's a transcription factor, FOXP3, that helps identify T regulatory cells, as well as the expression of CD25 and 127. Furthermore, my subsets of T regulatory cells, once I've defined them and found them, then I want to be able to look at naive and effector cells, as well as, for example, CD15S and what's thought of being as maybe more of a suppressive cell, or CD161, the more pro-inflammatory and cytokine-secreting regulatory T cells. So this defines my T regulatory subsets. But I also want to look at activation of the T regulatory cells. And within the activation, or to look at activation, from the literature and other sources, we know that there's a number of markers that we would want to include, such as PI6 and CD39 and CD147 and HLA-DR, all markers that are known to identify the activation state of different T cell subsets and T regulatory cells. But meanwhile, I also want to look at homing antigens. In this particular situation, I'm interested in looking at CD31, CD183, CD194, and CD196. And all of these markers are all different antigens that are of interest to me. So the problem is I have 12 detectors on this instrument, but I have 16 markers that I want to look at. Well, how do I accommodate all of these markers? Well, to do this, I can think about a different way of building my panel. So if I'm working with the BD Faxlier 12-color RUO instrument, I can basically use a modular flow cytometry approach to study Treg cells. I can think about it as if I'm going to build a panel that has an 8-color backbone to identify regulatory T cells, which is the population of interest, and define the major subsets. I can then also add in an additional 4 colors to get me to my 12-color limit on the system. And I can then also have a second tube, because I may not always want to look at all 16 markers simultaneously, and honestly, I kind of subset them into different buckets, and I can have a separate 4-color drop-in panel to define the rest of my cells of interest. And in this situation, it happens to be a panel that I can run whenever I'm looking at activation, and another panel that I can run when I'm looking at homing. And so this approach actually allows and enables a deeper characterization of the heterogeneous T regulatory populations. I can use 2 different panels focused on 2 different important aspects of Treg biology. And from working from a common core base, I can actually utilize high-dimensional analysis as well and kind of make some correlative comparisons, as we'll talk about later. So I can correlate the interplay between these markers and identify relevant subset and subpopulations in clusters. And all of this can be done in a simplified workflow. It also helps standardize. So if there's other people that want to look at other areas in the different aspects of the biology, they can just create their own drop-in panels. So how do we do this? Well, we have to kind of think about the panel design a little bit differently. So I have a 12-color instrument, but I want to build a panel that performs very well. I want a good core antigen backbone panel, and I also want to be able to add in my drop-in panel without having any major impact on my common backbone. So I have to think about it, or I like to think about it in a way of what fluorochromes do I need to save? And so when I think about that, I want to think about it that I basically need fluorochromes that will have minimal impact on the common core. So the backbone, I want to make sure that I can always resolve my core backbone panel and the specific subsets that I'm interested in. So a couple things about panel design in general is that moderate to bright fluorochromes will impact other fluorochromes less than very bright fluorochromes when co-expressed. Now, a lot of that has to do with something that I'll mention occasionally, but we won't go into the details of it, and it has to do with spillover spreading error and the loss of resolution. Of course, there's publications that are available, and if you've ever attended a BD Horizon tour, you're probably familiar with these principles. So we want to probably think about saving medium to bright fluorochromes. Also, the moderate to bright fluorochromes are a little more universal in their application. So if we have a bunch of very bright fluorochromes that we've saved and high antigen density is what we're looking at, I'm kind of not doing the best approach for panel design. I want to pair bright with dim antigens and dim fluorochromes with high antigen density markers. So if I choose something in the middle, I have a little bit more flexibility. Also, in general, the fluorochromes from the lowest wavelength and the highest wavelength lasers will generally have less impact on other laser lines. This has to do with cross-laser excitation. So when I look at my FACS Lyric instrument with a blue, violet, and red laser, I may want to think about saving fluorochromes off the violet and red lasers. So I also want to think about the fluorochromes that I'm saving. I want them to be impacted minimally from the common backbone. So if the backbone is impacting the ability to resolve my drop-in markers or my fluorochromes, well then it kind of defeats the purpose of having this modularity. So when I think about it, violet fluorochromes, again, in this situation, have minimal cross-laser excitation from the blue laser and the red laser. So again, I'm considering choosing violet fluorochromes or fluorochromes off the violet laser. Also, base fluorochromes have minimal impact on corresponding tandems. So what that means is if my common backbone is built of a lot of bases, they won't hurt my tandems if I use tandems as my drop-in markers. Now there is, however, a watch-out that my tandem fluorochromes can have residual spillover into the base. So this would be an example of PE and PE size 7. PE size 7 will have some spillover into the PE channel, and this could lead to some loss of resolution in PE. So we need to evaluate this in our specific assay. And of course, we need to evaluate the impact of our drop-ins, specifically our drop-in panel, on the backbone panel. So let's get started on some panel design. So on the left, you can see a list of antigens and the specificities. Those are what I call my common core backbone antigens. Those are the markers that I need to use to define my subsets of interest. On the right, you'll see a list of fluorochromes and the corresponding laser line that they're off of. Well, when we're thinking about fluorochromes that we want to save, we wanted to save fluorochromes that were probably off the violet laser and also off the red laser, and we might want to just save tandem fluorochromes. So what you'll see is that the fluorochromes are listed from BV421 on the top to FITC on the bottom with a gradation of orange, and that's meant to indicate fluorochrome brightness. Now, where we got that fluorochrome brightness, I'll talk about shortly, but in general, just think about this, that BV421 is probably, or is my brightest fluorochrome on my specific instrument, and FITC and BV510 are going to be my dimmer fluorochromes. So I want to save some tandem fluorochromes for drop-ins. And off the violet laser, I know that BV421 is a base, and BV510 is a base, and I have 3 tandems. I have BV605, BV711, and BV786. So let's save those for my drop-in. I also have the red laser. And with the red laser, we have APC, which is a base, and I have 2 tandems, APC R700 and APC size 7, or APC H7. So when I want to save another fluorochrome, I want to think about it to make sure that I have the best utility and versatility for my drop-ins. I already have a number of fluorochromes that are kind of moderately bright, so I'm just going to choose in this situation APC R700 because it's a little bit brighter fluorochrome. So without those fluorochromes being available, I now have my list of available fluorochromes to build my common backbone, the common core to my modular panel. So now we can go into a little bit about panel design considerations. Before we start to build this panel, what do we need to know? Well, as I mentioned, we want to know the fluorochrome brightness. Now, this is available from various resources, but you can also characterize the fluorochrome brightness on your specific instrument. And this can be very useful because various instruments have various performance characteristics and ability to resolve certain markers based off of how bright fluorochromes are on that specific instrument. So to do that, what you can do is you can take a common source for staining, such as whole blood, and what you want to do is use the identical clone against the same antigen. In this situation, CD4. Now, the reason we want to use CD4, it defines a very nice, distinct positive population in my lymphocytes, and I can gate on it very easily. It also, in the whole blood scenario, has a dim CD4 population, the monocytes. I can visually get an understanding of how well I can resolve a dimmer, lower density antigen on my system. And once I acquire all of these on my flow cytometer, I can do a calculation such as a stain index or a separation index. All of these calculations are available to find online at various resources, and even some software platforms have these calculations built in now. And I can use that stain index calculation as an indication of the relative brightness of my fluorochrome, since all factors are equal. So I can basically create a chart like this where I have BV421 ranked all the way down to my dimmer fluorochromes, Percy-P Sci-55, BV510, and FITC on the BD Faxileric instrument. Now when we look at this, this is relative fluorochrome brightness, and what you can see is that they're quite unique or quite different from FITC all the way up to BV421. However, when you look at the scale actually shows that there's really good resolution on most of these markers. They're considered very bright or they're performing very well and have a good dynamic range of resolution. So any one of these fluorochromes have a utility. It's a matter of using them appropriately. The next thing we want to know is about the antigen density. We want to pair bright fluorochromes with dim antigens or low density antigens, and then high density antigens with dimmer fluorochromes. So to do this, I often like to do a pilot experiment where I confirm the antigen expression level on the cell subset of interest. For example, you could say CD4 is high density antigen, and on lymphocytes that might be true, or specifically on T cells. But on monocytes, that may not be the case. There may be more of a mid-expression antigen. So you want to know what cells you're looking at, and we know that various cells express various levels of markers. So to do this, you can do a little preliminary experiment. And in this situation, I went ahead and looked at CD3, CD4, CD127, and CD25 to find my T regulatory cells. So from my CD4 positive T cells, I can look at a bivariate plot of CD25 versus CD127, and I can clearly identify my T regulatory cells. Now what you'll see is my T regulatory cells are very high for CD25. Meanwhile, the rest of the CD4 cells, there is an expression of CD25, but it's dimmer. So this is already telling me something about antigen expression. Now I can use a single fluorochrome, such as BV421, which has minimal impact on the panel and is a very clean antigen, and then look at the markers that I'm interested in evaluating and looking at their relative antigen density. And it's all relative, so to this particular cell population. You'll also notice I have CD45RA in there, and I'll talk about that in a minute of why that was included. But what we can see is if I gate on my T regulatory cells and then I look at the expression level of CD147, it would be considered a high expression level antigen, as well as CD15S. HLA-DR is more of a continuum, and PI16 would be maybe medium expression, and CD39 and CD161 is getting down to be kind of a lower expression. Now, I know that when I build this panel, there's going to be a lot of co-expression. CD4 is going to be co-expressed on my Tregs, as well as 127 and 25. And CD45RA is a high-density antigen as well, and I use it for subsetting my naive and effector regulatory cells, or T regulatory cells. So I want to understand a little bit about antigen expression and co-expression. And I mention that because I included CD45RA, and I want to know what that antigen expression looks like across a number of the markers that I'll be evaluating in my panel. So here in that panel, I have CD45RA on my y-axis plotted against all my other markers that are going to be in my panel. So for example, we have CD147 right here, which you can see there's a lot of co-expression of CD147 on both CD45RA positive and negative. Meanwhile, the rest of my markers actually have minimal co-expression. And this is actually important because when I start designing my panel, it tells me that CD45RA, I don't have to be too concerned about the co-expression of other markers in that particular fluorochrome impacting the resolution of other markers that I'm interested in looking at. For example, CD161 right here is another marker that I'm particularly interested in, and I can see that there's no co-expression in my CD161, CD45RA upper right quadrant in this particular plot. So now I've learned a little bit about my antigen co-expression and my antigen density and my fluorochrome brightness. So I can go back to my panel and start filling in some information. So as you can see, I have my configuration of my instrument listed with all the laser lines and the fluorochromes. And where it says antigens, on the fluorochromes that are going to be used for drop-in markers, I have just labeled them as drop-in, which tells me I can't use BV605, 711, BV786, or APCR700 for my common core antigen. I also listed the relative fluorochrome brightness. As you can see, I've saved some moderate to bright fluorochromes and also have something that could maybe be considered a very bright fluorochrome, APCR700. On the right, I have my core antigens listed, the ones that I'm interested in looking at in building my backbone, and their relative antigen density. And you can see I have a mixture of low, high, and medium. Also, for the characterization of my T regulatory cells, I have my activation panel here, and also my homing panel listed here, and the corresponding antigens. So now I want to build my panel. You can see that my antigens are shown in different gradations of gray. So how we actually identify our antigens and kind of characterize them is not necessarily always just high antigen, mid antigen density, and low antigen density. There's some publications out there that have actually helped us kind of characterize our antigens to put them into various buckets. For example, from the Roederer group, they talk about primary antigens. Primary antigens are those that are well characterized, identify broad sets of cells, and their expression is usually either on or off. So I could say CD3, CD15S, and CD4 kind of fall into that category here. And so they're a darker gray. I have secondary antigens that are also well characterized with relatively high density per cell, but their expression pattern is often a continuum. And an example would be CD45RA and CD127. And then we have the tertiary antigens that are expressed at low levels, and often you can also find things like transcription factors and things that are a little harder to resolve that will be put into that bucket. And the tertiary antigens, when I'm thinking about building a panel, are the ones that I'm really thinking about I want to protect them. I want to make sure that they're well resolved and anything that may impact them, I want to be mindful when I'm assigning fluorochromes to my other markers when I'm trying to make sure that I'm able to resolve these populations in the end. So when I look at a panel like this, I want to assign a fluorochrome to the antigen that I think is most critical for me for resolution in my panel. And in that situation, it's FOXP3. The FOXP3 is the transcription factor that helps define my T regulatory cells. And from previous experiments in the laboratory, and as well as looking at TDSs and evaluating the literature, I know that FOXP3 performs very well on Alexaflor 647. So that is my first choice for comparing my antigen with my fluorochrome, and that's off the red laser. Now I know I want to be able to resolve my T regulatory populations. FOXP3 is a part of that, as well as CD25 and CD127. Now CD25 is a tertiary antigen. I need to make sure I have really good resolution of that particular marker. So I'm going to put it on BV421, one of the brightest fluorochromes I have and one of the cleanest fluorochromes when it comes to spillover. I also want to take care of CD127 right away. So I'm going to pair CD127 with a different fluorochrome off a different laser line, Brilliant Blue 700. I know that Brilliant Blue 700 is a very bright antigen, and there's going to be no impact between Brilliant Blue 700, Alexaflor 647, and BV421 when it comes to spillover spreading to air and potential loss of resolution. So now when I start thinking about what I want to do next, I want to make sure that I start assigning co-expressed markers in a way that they shouldn't hurt any of these other fluorochromes. And so we'll start out with CD3. CD3 I know is a high antigen, high density antigen marker on my T cells. It is a primary antigen, and I'm going to put it on one of the dimmer fluorochromes, APC size 7. Now I know APC size 7 from familiarity with the fluorochrome that it may be impacted by other markers and other fluorochromes, but that's okay because CD3 resolves very well. I do have to be mindful of the fact that APC size 7 has some residual spillover into the APC channel, so I have to keep an eye on FOXP3, make sure we don't have any loss of resolution. The next marker that I want to look at is CD4. CD4 is co-expressed on everything, so specifically the T regulatory cells. So I want to put that on a fluorochrome that isn't going to impact other markers as well. And FITC is a good choice for that, especially because it's a bright primary antigen. So now I'm left with a few remaining markers, 161, 45RA, and 15S. So 161 I'm going to pair with PE. I'm running out of very bright fluorochromes, and so I want to pair it with something that I know will be able to resolve that antigen quite well. And then I want to make sure that 15S, since it's a primary antigen, is paired with one of my dimmer fluorochromes, and that leaves CD45RA PE size 7. Now 45RA can be highly expressed, certainly on naive cells, and so there could be residual spillover into the CD161 PE channel. But as you saw in my co-expression work earlier, there really is no co-expression of CD45RA and CD161. So that tells me I probably don't need to worry about that. So now this is what my panel looks like. I built my backbone panel, my drop-ins are left out, and now I need to be able to define my T regulatory cells, or I can define my T regulatory cells as CD4 positive, 127 low, 25 high, FOXP3 positive. I can identify my naive and effector cells based off of CD45RA, my suppressive CD15S cells, and my pro-inflammatory cytokine secreting cells, CD161. And I also have my drop-in activation and my drop-in homing panel. As you can see, my activation panel, I just assigned fluorochromes based off of known expression patterns of my relative or my specific antigens on my cell population of interest, as well as fluorochromes. And I'm not going to go into the details of the assignment of these fluorochromes, and instead we're going to go ahead and start looking at our data and see how our panel's performing. So what I've basically done is created a backbone panel that has a lot of flexibility. I have my drop-in activation panel, my drop-in homing panel, and in theory, I could build more drop-in panels that looked at, for example, maturation or proliferation, stability, whatever it is that my laboratory is interested in, but we're all working from a common backbone to define our T regulatory cells. So our outline of how we're going to look at our data is we're going to look at our results and look at the impact of the 4-color panel on the backbone. We're also going to evaluate homing and activation markers by conventional methods, as well as a high-dimensional data analysis of our T regulatory cells. So how did our panel fare? Well, we can take a look at our first donor here, and we want to look at the ability to resolve our CD3, CD4 positive T cells, and of course, not surprising, it works or looks very good. I'm able to easily define my CD4 T cells, and then I want to look at and be able to define my T regulatory cells based off a CD25 high, CD127 low expression, and of those, identify my FOXP3 positive T regs. From there, I can look at, within the FOXP3 positive T regulatory cells, my CD45RA and CD15S expression as well as CD161. And you can see that I get very nice resolution of all these populations. This is the backbone panel alone. Well, what if I drop in one of my panels, one of my drop-in panels? In this situation, I dropped in my homing panel. And you can see that if I look at my populations of interest that I want to look at, I'm still easily able to resolve all of my T regulatory cells, my FOXP3 positive cells, as well as my subsets that I'm interested in. And I want to do the same thing with my activation panel. And you can see, once again, that dropping in of my activation panel did not impact my backbone at all. So what we know now is that we've designed some panels that are very conducive to drop-in panels to be able to evaluate different areas of T regulatory biology. Now, the areas where I was a little concerned that I wanted to keep an eye on is that we can see a little bit of spreading air. Maybe we have a little wider area in my CD45RA PE-Cy7 negative cells, but that's all right because there isn't much co-expression that I'm necessarily interested in looking at with those particular markers. So let's talk a little bit about helper T cells and T regulatory subsets, because we're actually evaluating these different subsets by looking at these different activation and homing markers. So we're all very familiar with helper T cells. So helper T cells can be broken down into naive T cells, our Th1, Th2, and Th17 and Th22 cells, and as well as Th9 and our T follicular helper cells. All of them are very important in the immune system and immune responses, ranging from responses to infection to autoimmunity. And interestingly, there's corresponding T regulatory subsets. So we have various markers that define those T helper cells, and you'll notice the CD183, 194, 196, those are actually a part of my homing panel. So when I look at my mature and effector T regs, you can see that there's changes in expression. For example, mature naive T regs start out CD45RA positive, and they go to CD45RA negative. CD15S and CD161 and PI6 all go up. CD147 goes up. You have HLA-DR expression. We also are able to look at our recent thymic emigrant T regulatory cells based off of CD31 expression. We also have induced T regulatory cells that upregulate FOXP3 from the CD4 T cell subset, ones that haven't developed in the thymus. And then we also have our TH-like T regulatory cells, our TH1 and TH2-like, TH17-like, and TH22-like, and their corresponding expression of homing markers. and where they may have roles in functional biology. So let's go ahead and actually take a look at our panel a little bit. So with our T regulatory cells gating on our FOXP3 positive Tregs, we can use the 4-color homing drop-in panel, and that helps identify TH-like subsets within the Treg population, as well as recent thymic emigrants. So we have our CD45RA expression, and we want to look at the CD45RA positive cells. Okay? From there, we can look at our TH1-like cells for CD183 expression, and our CD183 negative cells, we can look at CD194 and CD196 expression to define our TH2-like and our TH17, TH22-like T regulatory cells. And we can also take the CD45RA naive, mature T cells and then look for CD31 expression for our recent thymic emigrants. And we can compare this to conventional T cells. So as you can see, in our same panel, we gated on just CD4 positive T cells, excluding FOXP3 T regulatory cells. And you can see our corresponding TH1, TH2 subsets, and our TH17 and TH22-like cells. So let's go ahead and evaluate the activation markers on our Tregs using conventional approaches, similar to what we just did earlier, where we can gate on our different populations and evaluate their expression. So we have CD15S versus CD45RA, and now we can gate on the different subsets and specifically look at what may be, or is called maybe our more suppressive T regulatory cells, and then those who are naive, mature, and effector T regulatory cells, and look at the expression of various markers across various donors. So here we have one donor where we're looking at the expression of FOXP3 in the upper left-hand corner, and you can see that there's different levels of expression of FOXP3 even within these 3 subsets that we're looking at from the 45RA versus 15S gate. There's also significant differences in HLA-DR expression, PI16, CD147 is positive for all 3 subsets with varying levels of expression. And interestingly, it kind of looks like the expression of FOXP3 similarly, as well as CD39 and CD161. Now we can go ahead and look at pools of donors where we did this with 3 different donors, and we can see that FOXP3 expression is rather consistent against a large number of donors. However, we can look at other markers that seem quite variable across donors. For example, HLA-DR within our CD15S positive population is quite variable. And the same could be true or said about CD39. And if we look at it a little closely in our bivariate plots, if we look at donor 1 and donor 2 and donor 3 for CD39 expression, you can see that the expression is quite variable when we look at it versus CD15S. And we can plot this based off of where there's the relative percentage of cells that are in the various quadrants using our conventional analysis approaches. And you can also see that, for example, in this situation, there's a unique difference for CD39, which we have a reference here for a paper that actually discusses some of those variabilities for CD39 expression that you see within donors. So all of this works very well for us to characterize our T regulatory subsets. We can define the subsets, our panels are performing very well, and of course we still have the flexibility of adding in other types of panels as well. But we can also think about looking at our data in a computational, high-dimensional data analysis way. So conventional flow cytometry analysis is often done iteratively, plotting cells as bivariate plots and manual gating at each step. There's some drawbacks. It can be subjective. There can be variability. Who's setting those gates? Will everyone set those gates the same, especially when you don't have the best resolution of your markers? What is the reproducibility? And then also the gating strategy and the order becomes critical. So do you go FOXP3 positive to define your Tregs Well, you could have induced Tregs versus if you want to look for thymic-derived T regulatory cells. And then some of these can be influenced by the ways you're actually gating. CD25 high versus CD127 low. Is that a better approach? And those gating strategies and the shape and boundaries of the gates will be variable. You also have a limited view of the data. It's obscured by analysis. You have an obscured analysis of how the different markers play together. And when you get very large data sets, it can be actually quite time-consuming, as you'll have numerous n by n bivariate plots. And this grows as your panels get larger. So you can look at your data in the computational flow cytometry approach. So the analysis is based on sound mathematical principles that has been described in a number of recent articles. And you basically can assess all of your markers simultaneously. It's objective and highly reproducible. But you have to understand that it's a visualization tool, and you still have to understand what it is that you're seeing. And we'll go through some some computational flow analysis of these two different panels. And as we know, our traditional approach of analysis identified the different subsets. However, let's see what we can do with a computational analysis approach. So we'll be looking at a couple different high-dimensional data analysis and visualization tools. The first one is going to be dimensionality reduction algorithms. And the goal is to preserve the main structure in the data while projecting high-dimensional data in a lower dimension for visualization. And this example is, maybe you've heard of Tyznik, T-distributed stochastic neighbor embedding. Well, obviously, I'm just going to sit and say Tyznik over and over because that's a mouthful. But it aims to preserve the similarity in the original high-dimensional data that you put into the system. Now, we're going to use what's called a Barnes-Hutt implementation of the Tyznik algorithm. That's done in MATLAB. But you can use whatever software program you choose to, and there's even articles out there guiding you through the appropriate utility or use of these tools for visualization of your data. We also have some clustering-based techniques that we'll look at. And they group cell types and clusters and visualize in a lower dimensional space how similar the clusters are. And so specifically, we'll be looking at Phenograph from MATLAB as well as XShift. And these are just, again, different ways to visualize your data and basically try to understand and reveal some of the heterogeneity within the T regulatory subset that we were analyzing, that we were actually maybe just interested in looking at some homing and activation markers, but actually turn out to be quite heterogeneous. So here is an example of a BISNI, or basically what's going to be a Tyznik, of our activation marker panel. And so we're going to go through where we have our common backbone panel along with our 4-color activation drop-in. And this is looking at one specific donor. Now, to do these types of analysis, you have to do a subsample of cells. Even though you acquired 1 million cells, you have to do a subsampling of them. And so in this situation, we subsampled 30,000 lymphocytes. And so when we put it into the algorithm processes all of the information simultaneously, looking at the expression of all of the proteins on all of the individual cells, and tries to make it so that we can kind of comprehend it in a lower dimension visualization. So when you run an analysis like this, cells that are similar in their protein expression pattern will be located closely together in the Tyznik map. So what you have to understand is what we're looking at here is our lymphocytes on this particular donor. And we ran the algorithm, and what it did is it grouped clusters of cells based off of their protein expression, if it's similar. And then we overlaid all of those lymphocytes with just the FOXP3 expression. So now what you can see is there's a hot spot of where FOXP3 is. So I can use that and say, those must be my T regulatory cells. Now all of these other clusters and groups, I don't know what they are right now. All I know is they are probably not Tregs because they don't have FOXP3 expression there. So the next slide is going to get very busy, and so don't get too overwhelmed. I'll walk you through it a little bit. But we take this exact same Tyznik plot, and we want to investigate it a little bit further. Instead of just looking at FOXP3 on the lymphocytes, let's look at other things. Now you can see that the Tregs themselves are kind of spread out. So let's go over into the upper right-hand corner to begin with. So we're looking at the same Tyznik from donor 7720 and looking at CD3 expression. Now we see all of those red clusters in the right-hand corner here, okay? So wherever it's red, those are going to be most likely a T cell. And so we can maybe start to make some conclusions about the data, about what other clusters could be, but I don't know specifically what they are. I could say some of them are probably B cells, some of them are probably the non-CD3 positive or the non-CD3 expressing cells. They could be my NK cells or my innate cells, my dendritic cells. But I don't know because I don't have markers to define them. All I know is they're placed in a cluster somewhere else that's blue, for example. So then if I overlay just my CD4 expression, as you can see right below it, you can now see that, well, those are my CD4 T cells. All right, well that's encouraging because I know my T regulatory cells on my far left-hand side, where my FOXP3 expression is, falls within the cells that are all red. So that tells me I'm on top of my CD4 positive T cells and CD3 positive T cells, and those are the cells that express FOXP3. You can also take a look across all of these other markers, which we'll work through, and we can find our T regulatory cells, as just showed up in the red circle, that they're CD127 dim. If I overlay the expression of CD127, you can see this area is light blue, or a lighter color. Meanwhile, my CD25 expression is red hot, right where the Tregs are. And we know that about our biology, correct? We know when we conventionally gate on our T regulatory cells, they're CD3, CD4 positive, FOXP3 positive, 25 bright, 127 dim. I can also look at the CD45RA expression. You can see that there's a continuum of expression across the larger CD4 group. So I'm now looking at the CD45RA plot. And you can see that there's a continuum of CD45RA expression, which we expect on all CD4 T cells. You also see the same pattern across the CD4 negative cells, which is actually probably our CD8 T cells, but I don't have a CD8 marker to tell me that. And I see the same continuum across the T regulatory cells. All right? Meanwhile, I noticed that there's a bright red population of HLA-DR positive cells in the next plot over. And so when we think about how Tyznik plots are built and how they're kind of visualizing the data, it's maybe not too surprising that whatever is HLA-DR positive is pulled so far away from all the other lymphocytes that I'm looking at because there's really no other HLA-DR expression there. However, those that are expressing HLA-DR, as we can see, are a little bit closer to that red clump. So we know that our HLA-DR expression is actually differentially expressed in different subsets of Tregs, as well as CD15S. And I know that CD147 expression, actually the expression pattern is the opposite of CD45RA. And I can see that when I'm looking at my T regulatory cells right now, as well as CD39, PI16, and CD161. So before we want to go into really drilling into the FOXP3 positive population, though, as I mentioned, the Tyznik plots will actually take all the information and recalculate everything based off of the relative expression of proteins when it's being ran at that individual time. So in this example, as I just put up, we have another donor that we wanted to look at. This is donor 5025, doing the same thing of doing a random sampling of the lymphocytes and looking at our activation markers. And what you'll notice is that this Tyznik plot looks different than the previous one, and that is to be expected. However, if we go to the upper right-hand corner, once again, we see that there's an overlay of the CD3, which defines my T cells, and we also have an overlay of CD4, where we can see what our CD4 positive T cells are, and we can maybe infer where our CD8 T cells are. And then we come to the upper left, where we look for FOXP3 expression, and those define our Tregs. And they also have the same pattern when I overlay 127 and CD25, where they're 127 low, CD25 bright. And interestingly, in this particular Tyznik run, the HLA-DR positive cells, just in the broader lymphocyte population, are pulled to the left. They're in a different location. And it kind of shifts where all the different cells go accordingly based off of all of the proteins. So each time you run these plots, they won't look identical. But you have to go through and then look at the markers that you're interested. For example, just as we saw in the other donor, even though it looks different, the CD45RA expression across the FOXP3 positive T regulatory cells, there's a continuum as we would expect. And that continuum is actually the opposite for the CD147 expression. Once again, something that we would expect and we saw in the other donor as well. Now, there's other types of analyses that you can do where that we talked about, such as X-shift. That's a cluster profile analysis, and we can use this to reveal distinct Treg cell clusters. And for example, we're again going to look at lymphocytes here, and then we can look across all of the different donors, and what you can see is that we defined various clusters of and patterns of expression across our various donors. Now, what you'll notice is that the clusters are actually kind of similar across all the donors. However, remember what we talked about for CD39 early on, where there's one donor that had a slightly different CD39 expression in our conventional analysis? Well, here we can see that donor 7720, when we want to look at the different clusters that were found, you can see that the different donors have different patterns. And this is just revealed from an automated computational analysis looking at it across various donors. So there's striking similarities in these clusters, but you can also use this to try to find differences or uniquenesses across the different donors that you're evaluating. So we now want to actually drill in deeper into the FOXP3 positive cells. And so we previously had looked at all of the lymphocytes, but we're now going to go ahead and look at specifically donor 7720 and look at the FOXP3 positive population only. We're going to do a random sampling of 30,000 cells within the FOXP3 positive population, and then run our Tyznik or Biosciences plot. Now, what you can see is we're only displaying the FOXP3 positive T regulatory cells in this particular plot. And then you'll look at it and say, hey, Aaron, why is it blue? There's no FOXP3 expression. Well, the important thing to remember is that they're all FOXP3 positive when we first gated on them, and within this group, this is the relative expression of FOXP3 within that population. So some are what we would say are low expressors, even though they're positive. And then you have a continuum or an area where those actually have much higher expression in the lower right-hand corner. And so I'd encourage you to keep an eye in this lower right-hand corner because it's going to tell us a little bit about the heterogeneity of these T regulatory cells. Because these that are FOXP3 positive, are FOXP3 high, I should say, they're all FOXP3 positive, have different levels of expression of other markers. So we can go ahead and take a look at our FOXP3 positive cells and then look for CD4 positive T cells. And you can see when we did our gating that there's some blue cells. So there's some gate or cells that fell into our gate that were not all CD4 positive T cells. But we want to look at this FOXP3 high population here, along with other things as well. So when we look at FOXP3 high expression, we can see that it actually correlates with the CD25 bright cells. So the high antigen, as we saw CD25 within the T regulatory subset had a higher expression level in one subset, and then maybe the 127 brighter population had a lower level of CD25. And you can see that here where we would say these are probably our CD127 higher populations, that those are actually the CD25 dimmer cells. We can also look at our expression pattern of our naive and effector cells. And we know that CD45RA is expressed in a continuum. And so our naive cells are nicely identified here. And then we can look at how that expression correlates with CD147. Now you remember from our original panel building exercise where we looked at co-expression, CD45RA co-expressed with CD147. So it's not surprising that the naive cells that we see here where they're for 45RA expression that are very red also have some expression of CD147. However, the effector cells have much higher expression, and that is actually shown here where CD147 is more red when we look at this heat map overlay. If we continue to look at the FOXP3 bright cells, we can see at the very, very tip of, on the bottom here of CD147, that those cells are HLA-DR bright. And they're also CD15S positive. We can also see that they're positive for CD39, but they really don't have CD161 expression. In fact, CD161 seems to have its own unique expression pattern all by itself. It's completely pulled away from all the other FOXP3 positive T regulatory cells. So these are the types of things that this analysis can help you with. It can help you visualize your data in an unbiased way. Now it's hard to maybe gate on these populations, so conventional analysis is still very important. We know that CD161, we could go ahead and look at these cells even further and say that, well, some of them are CD15S positive. They have a continual expression of CD147, and they also have a population that is CD127 high. So those, again, are different ways of being able to kind of analyze your data using a computational analysis approach. We can now actually move on and look at our homing markers. So as we mentioned earlier, we had a number of markers that were our homing markers to define our T regulatory subset. Now we've looked at the activation, and this is just looking at a random sampling of 3 donors that we have in-house that we just draw randomly. Well, when we want to look at the homing markers, we want to look at another type of computational analysis. So we can go ahead and start out with a BISNI analysis of our lymphocytes, and we again look at donor 7720 and do a random sampling. And in this situation, we can now see that when we look at our FOXP3 expression, they're actually kind of spread out a little bit. They didn't all cluster together. And that may seem surprising, but when we think about it, when we've added in these new homing markers, it kind of clustered all of the cells together, all the lymphocytes, the CD4 T cells, the CD8 T cells. It's taking all of them and kind of putting them near each other based off of their protein expression. And so the FOXP3 or the Treg markers may not be more dominant in terms of the particular proteins that were being analyzed in this panel. But they're within proximity of each other. But when we analyze now our lymphocytes population for expression, we have to know where our Tregs are. So we do the same thing where we have in the right our CD3 and CD4 analysis to show that we're on our Tregs, and then we have our FOXP3 expression. And as you can see, even though they're spread across 3 different areas, we have CD127 low and CD25 bright. We also have different expression levels of CD45RA, as well as unique expression of these other markers, CD183, 194, 196. And we'll go into those markers a little bit further, because we can now take a closer look into specifically the FOXP3 positive cells. So this is a random sampling of the FOXP3 positive Tregs, and then look at the homing markers. Again, we can see that there's the CD127 expression and the CD25 expression. However, the proteins that are in here right now really clustered the cells differently than our activation panel. And again, that's something that we have to be aware of for the way this is used as a visualization tool. So we can see that our FOXP3 low expressors are the ones that have lower expression of CD25, so that's consistent. We also have naive cells being clustered away from our effector cells for CD45RA expression. But we also see what is Th1-like T regulatory cells based off of CD183 expression. We also have what is Th2-like cells. And within this larger group, we have our Th17, Th22-like T regulatory cells, and also a unique subset within our CD161 expressors. They're pulled out away from all of the other T regulatory cells. We also can clearly identify what would be our recent thymic emigrants based off of our CD31 expression. And essentially, using an approach like this, we've basically been able to further resolve and identify the heterogeneity within the FOXP3 positive T regulatory cell population. And this is done through a computational analysis. This is without gating, subjectively trying to identify where your population is. In this particular example, though, we sampled in on the FOXP3 positive cells only, and then looked at our homing markers in that situation. So we'll look at another type of analysis, as I mentioned, the phenograph analysis. And so this is where we pooled together all of our T regulatory samples for our homing markers. We concatenated them, basically. And so we pulled the FOXP3 positive Tregs, regulatory cells, from the 3 donors, and phenotypically similar clusters of cells will be represented as what we call neighborhoods of cells. So to do this, we put it into the algorithm, and then it identifies, basically from these pooled T regulatory cells, 14 different clusters within the T regulatory compartment. So that tells us a little bit about the heterogeneity of the T regulatory cells in this homing panel. Now we can then go ahead and take these pooled T regulatory cells and run a BISNI and algorithm, and then identify our 14 different clusters of interest. So this is just color-coding all of the different clusters that were identified. Now, we can then take this a step further And you know how we had previously kind of overlaid our different markers, our different antigens that we're interested in? We can now compare this to our phenograph clusters. So I'll draw your attention to CD183 here, and this is our Th1-like T regulatory cells. And then if you want to go ahead and follow your eyes over to our phenograph cluster, for example, right here, we can see that the CD183 clusters actually fall within some of the unique clusters that were pulled out in phenograph. And so all of these algorithms are all kind of working together, telling us more about our cells of interest and how they may be subsetted in a high-dimensional space. Because no longer are we just hierarchically gating down into different subsets. It's taking all of that information and using all of the markers that we wouldn't even think about, that actually may be important for identifying these cells of interest. We also can look at our Th2-like T regulatory cells based off the CD196 expression, and when we look at our phenograph clusters, again, they show up as this more pinkish population here in our phenograph clusters. So we also can see that we have our Th17 and 22-like T regulatory cells, and all of those fall into all sorts of different clusters within themselves. And so we also can dig in a little bit deeper and see that our CD161 is very, very distinct in its expression, and again falls in within one of the the clusters, again, that was identified from our phenograph clustering algorithm. We can also see that we have our recent thymic emigrants. The phenograph identified our recent thymic emigrants as this darker black population, saying that those are a unique population across all three donors, because again, those phenograph clusters were generated by pooling our donors. And we're now just looking at one specific donor and see how it fits into this pooled population. So now you could do a comparative analysis. How is this donor compared to a pooled sample of donors? And again, we've further dug in and been able to really resolve the heterogeneity within the FOXP3 positive T regulatory cells. So in the end, I hope you can appreciate the utility of a modular flow cytometry approach like this, where you can go ahead and build an 8-color backbone panel, or whatever is necessary. If you need 10 colors to define your population of interest, or if it only requires 4 markers to define it, to build a base backbone panel, and then add in your activation markers, things that you're interested in. For example, here we did a 4-color activation panel. We also did a 4-color homing panel. Again, this was for T regulatory cells, but it could be applicable to any type of panel that your particular laboratory is running routinely. And again, there's a tremendous amount of flexibility. I know I can clearly resolve my core populations of interest from my backbone panel, and I can add in other drop-in panels, whether I wanted to look at cytokine expression, whether I want to look at maturation. Maybe I want to start looking at different donors and choose different markers that have to do with development. Really, the key is using a modular flow cytometry approach. You can have a lot of flexibility, and especially, as we understand, most laboratories have certain populations of cells that they're interested in looking at. So in summary, overall, these modular 12-color panels enable identification and deeper phenotypic characterization of Tregs. It enables simultaneous assessment of multiple markers. We can look at the correlation of the markers, the interplay between them, and reveals biologically relevant subsets within the rather heterogeneous T regulatory population. No longer do we think these cells are just vanilla. They are extremely heterogeneous, And with these computational tools, we certainly are able to appreciate just how heterogeneous they are. We know that there's a subset of Tregs that express high levels of FOXP3, 147, HLA-DR, CD39, PI16, and CD15S. We identify distinct Treg clusters and T effector-like subsets. And this offers flexibilities by the use of drop-ins, essentially reducing workflow by using standardized tubes that you could then go ahead and add your drop-ins accordingly. So really the take-home message from this webinar, though, is that I want you to appreciate the fact that T regulatory biology is complex. And this is actually spreading into all areas of the traditional cells that we've been evaluating, whether it's NK cells, whether it's dendritic cells. So understanding Treg biology at a deeper level, of course, is critical. And using panels like this and using these types of approaches can allow you to interrogate deeply within these populations. And using newer, higher dimensional analysis tools, you can better study and visualize your cells of interest. And these were all done on a BD Faxileric 12-color flow cytometer, again using a modular 12-color panel approach.
Finally, I'd like to let people know that I work with an incredibly talented team here at BD, and I want to extend the acknowledgments to all of the people that actually enabled this webinar and help with content. Specifically, Nihan Kara has been significantly involved in generating the content that you've seen today, as well as Mirko Corselli in the formulation of the idea of a modular T-regulatory panel. We also have extremely well-versed computational biologists here as well, that specifically being Joe Trotter, and some of our spectacular scientists, Alan Stahl and Noel Warner, had significant input in the scientific aspect of panel design as well as T-regulatory biology. I also would like to acknowledge Saras Saxena, who is basically the director of or associate director of our applications element, and is incredibly critical for us to be able to generate this type of data and share it with you, and as well as some an acknowledgement to Margaret Inokuma for some of the early development of the different T regulatory populations that we chose to evaluate. So I'd like to thank everyone for their attendance, and this concludes our modular flow cytometry approach to examine regulatory T-cell biology, specifically activation and homing markers.
Thank you. And as a reminder, ladies and gentlemen, if you would like to submit questions via the web, please click the green Q&A icon on the lower left-hand corner of your screen, type your questions in the open area, and click Submit.
So we'll begin our Q&A session now, but before we do, I just want to ask if you could take a moment to complete a poll that will be appearing on your screen shortly. We did have a few questions that came through from the audience. So, Erin, the first one I'll begin with is, do we need to add a viability stain to the panels?
A viability stain is an excellent question, and of course that relates to your specific sample that you're working with. Here in this particular panel, in the data that was shown today, I was working with fresh whole blood that had been drawn and then analyzed within a few hours. And in that situation, whole blood has an extremely high viability, where I'm confident in the staining of my samples, where I'm probably working with 98, 99% viable cells. Now, of course, that depends on your specific application, because something like a processed tissue or even PBMCs or blood that has been sitting around for a couple days, you'll start to see cell death, and we want to make sure that our data is as accurate as possible. In that situation, you can consider adding a viability dye, assuming that you have room within your instrument based on your configuration to add that additional marker.
Great. Next question. When looking at my data using a computation analysis such as Tyznik, is there an easy way to correlate it to traditional gating analysis?
That is a great question, and unfortunately at this very moment I would say the answer is not yet, but there is a strong effort from many different people in the field of flow cytometry that are trying to address this, to enable an easy translation from what you see in an analysis, a computational analysis like Tyznik, and then identifying it back to what gates are necessary to find those populations. There's applications such as Gate Finder and so forth that are trying to connect all these pieces together. So as you saw when I presented the data here, I showed you a traditional hierarchical gating, and then we use that as just our reference. So we can go back and overlay different populations and make assumptions that the population I'm probably seeing through this visualization tool, such as a Tyznik plot, corresponds to a specific population in my gating strategy. And that allows me to actually start dissecting it, my traditional gating strategy, in different ways, because I can see the data in a different way, and I may have a bias in excluding a marker that actually may be important and can tell me more information about the cells I'm interested in. Okay, next question. I have an 18-color instrument. If I were to use a modular approach to panel design, would I just save the same fluorochromes for my drop-in panels? That's another great question. So if you have an 18-color instrument and you have the corresponding 12-color markers on there, you could choose to save the same fluorochromes. However, you could also say that you know the 12 colors work together very well, and so, but you know, if you have an 18-color instrument, you may have additional capabilities. So in this situation, I was working with a Fax Lyric that has 12 detectors of a blue, a violet, and a red laser. But for example, an 18-color instrument, you may have a UV laser or a yellow-green laser. And all of these have other fluorochromes that are extremely valuable. And you may choose to use something such as BUV 395, a very clean fluorochrome that could be both dropped into your base panel or actually used and saved as one of your drop-in panels, for your drop-in panel. So really it depends on your particular instrument and how you're trying to build this panel and saving the things that are important. Last question. Do I need to worry about my base panel impacting my drop panel resolution? That must be a question about your drop-in panel. So does your base panel sometimes impact your drop-in panel? And absolutely. So that is of concern, and so we thought about it from a perspective of what do we want to have drop-in fluorochromes that are rather diverse in utility. So something that's bright or moderately bright, and then try to put them in a particular laser choices. But you could also design this panel in another approach where I want to save my best, cleanest fluorochromes so that they're going to be on my drop-in panel, and so I know my base panel will not impact them in any way. And of course, there's multiple approaches to panel design, and it really depends on your specific application and your instrument capabilities. I know that BV421, FITC, and APC are very clean fluorochromes, and I could have just saved those as my drop-in panels. But, you know, my particular panel design for my application, I needed to actually use those on other markers on my base. So those are, but it's a definitely good question, and it's something that needs to be considered, and you can consider that when you're designing your own modular panels. Thanks so much, Aaron. Thank you so much for an excellent presentation. And just a couple of reminders to the audience.
So you can earn PACE credits for this webinar. So look out for an email within the next day or so for a survey to earn PACE credits. And the webinar recording will be available online in about a week's time. And then thirdly, we will be having another webinar in September, so look out for that email. And we really appreciate your attendance today. Thank you so much for participating, and again, thanks to Aaron and the team here. We'll sign off now and enjoy the rest of your day.
Thank you, presenters, and thank you to all of our attendees for joining us. We hope that you found today's webcast presentation informative. This will conclude the program. You may log off at this time and have a wonderful day.